Hi everyone, I'm Fushen Zhou from Anhui Medical University of China. Today I'm going to talk about a toxic analysis revealed a widespread increase in chromatin accessibility in psoriasis. Psoriasis is a common chronic inflammatory skin disease that is characterized by thickened, scaly, and plaque lesions. There are four types listed here. Today, we mainly talk about psoriasis vulgaris. The key histopathology of psoriasis including hyperproliferation of epidermal keratinocyte, infiltration of immune cells such as neutrophil T lymphocyte, and among others. Psoriasis heritability is about 76% in Chinese population. To dissect genetic background of psoriasis, we previously performed psoriasis GWAS in more than 10,000 individuals and identified about 40 susceptible loci, including IL-12B, IL-23R, LCE3C, and MHC. Most of these loci have been confirmed by other groups, but also most susceptible loci located within the non-coding regions. That means they might be regulatory rather than directly impact gene functions. DNA methylation studies revealed more than 1,500 loci that contribute to disease development, and also you can see here most of these loci located in intergenic gene body or open seas, indicating they might be also regulatory too. We also identify more than 1,000 differentially expressed genes by transcriptome sequencing in the psoriasis and normal skin tissues. As over 95% of genetic or DNA methylation signals appear to be regulator, chromatin structure can place regulatory signals many thousands of BP away from the key genes being regulated. Tools are needed to interpret disease association signals in regulatory context. Human genomes are compacted hundreds of thousands fold in chromatin, and only the active regulatory elements are accessible by the cell's machinery, such as transcription factor. ATAC quantifies DNA accessibility through the use of transportase enzyme that insect sequencing adapter at this accessible chromatin site. ATAC enables the genome-wide proliferation of transcription factor binding events that alter the gene expression pattern and give a cell its identity. We carried out ATAC for 21 psoriatic skins or PP, 9 psoriatic non-lesion skins or PN, and 15 normal skin tissues or NN. All these samples have been assayed for methylation and expression in our previous study. After filtering raw sequencing data, we found most samples obtained a characteristic fragment size distribution with clear nucleus periodicity. A total of nearly 1 million consensus peaks were identified. Comparing with DNA's data of many primary tissues or cell lines from the ENCODE project, more than 40% of our accessible peaks overlapped with the keratinocyte or skin tissues, suggesting the reliability of our sequencing data. Here we list some conservative genes for presentation. At a just FDR p-value at 0.01, we find 22,000 and 50,000 peaks for PP versus PN and PP versus NN comparisons respectively. Of these, more than 90% showed increased accessibility in PP samples. Both comparisons also exhibit strong consistent fold changes. That means PP and NN groups share the similar accessible patterns, while distinct from the PP groups. Principal component analysis based on all 1 million consensus peaks showed that most of PP samples were clearly separated from the other two groups. Further confirming our findings, we applied a more stringent cutoff, thus fold change greater than 2 and FDR less than 0.01, and identify nearly 5,000 peaks, hereafter denoted as differentially accessible regions or DARs. S108 family of genes harbored multiple peaks associated with significant increased accessibility in the PP samples. Gene ontology and pathway analysis also showed an enrichment in related processes, such as epidermis development, adherent junction, and TNF signaling pathway. DNA methylation and toxic datasets allowed us to explore the underlying connections for all nearly 5,000 DARs. 1,200 contain a DNA methylation, and 90 of them were associated with psoriasis, that is 7.3%, but only 0.6% of genome-wide ATAC-associated CpGs were linked with psoriasis. That means psoriasis-associated DNA methylation was significantly enriched in accessible regions. We also observed decreased DNA methylation pattern in these DARs. Among the DAR-linked genes, 632 and 274 were significantly up and downregulated in PP samples respectively. Most of these genes are more open accessible, especially for those located within the promoters. These upregulated genes significantly enriched in regulation of response to stress, cell death, programmed cell death, codified envelope, and immune response. To explore whether psoriasis susceptibility loci linked to DARs, we analyzed 531 psoriasis-associated SNPs downloaded from the GWAS catalog and identified 2 of them that overlapped in our DARs. These 2 SNPs located near PSMA6, NF-κB-IA, and OS2 genes, and all these 3 genes showed upregulation in PP samples. Notably, this analysis also told us only a small fraction of psoriasis variants located to DARs in skin tissues. Analysis of chromatin accessibility via ATAC-seq not only identify regulatory regions for transcription, but also can infer transcription factor activity within them. We found that DARs were highly enriched in motif in transcription AP1 transcription factor, and FROM1, also called FORCE1, ranked first. A higher motif score would represent a strong affinity for transcription factor binding, and you can see here FROM1 motif score is much higher in DARs than those from the less stringent 2 groups. And FROM1 also showed increased expression in PP samples. In our transcriptome sequencing data, we extract all the AP1 gene family, and we can see FROM1 expression was increased by more than 8-fold in PP samples compared with that in the other 2 groups. The average baseline abundance of gene B was marked higher, whereas the fold change in PP was much smaller, and we observed a similar expression pattern in a public dataset. We thus select FROM1 for further analysis. Immunohistochemical and immunofluorescence analysis observed that FROM1 expression was upregulated in cells from the basal layer and looks like more pronounced in subbasal layer of the psoriatic skin tissues. We then overexpressed FROM1 in NHEK cells and performed RNA-seq. We found that some psoriasis-associated genes were upregulated in overexpressed cells, including CXCL1, IL-23A, interleukin-6, NF-κB. That means FROM1 might be involved in the immune response in epidermal keratinocyte. Take a close look. We found that AIM2 genes was among the topmost DARs that contain the FROM1 binding motif. AIM2 is an important component of AIM2 inflammation that play key roles in regulating innate immunity. AIM2 was more accessible in PP samples and contained DNA methylation CG07195224. Accessible peaks was strongly negatively correlated with the methylation level of this loci, but positively correlated with AM2 mRNA expression. We suspected that AIM2 might be directly targeted by FRAM1. We observed that expression of FRAM1 significantly activated luciferase expression in cells transfected with the wild-type AIM2 promoter, but not with the mutant promoter. Analysis of public ChIP-seq dataset also suggests that FRAM1 binding to the AIM2 promoter in epithelial cells. These data indicate that FRAM1 upregulation contributes to psoriasis possibly through the AIM2 inflammation. In summary, we identified nearly 1 million accessible DNA elements, further expanding the repertoire of regulatory elements of human skin tissues, and the nearly 5,000 psoriasis-associated DNRs provide obvious information on the susceptibility, methylation, and also mechanisms of the disease. FRAM1 might regulate skin immunity through AIM2 inflammation. I would like to thank all these people make great contribution to manuscript preparation, statistical analysis, and biological experiment. I would also thank all the patients involved in this project and all the clinic doctors and students in our group. And thanks for your listening.